Hi everyone, I'm William, and I'll be talking about some joint work that I did with an undergrad, Karen Deal, who is an undergrad at University of Alberta, and this was in collaboration with my postdoc mentor from years ago, Griffin Weber. So the basic idea that we're going to be talking about today here is a new web app that I developed called SlowMoMan, and so all of us here know biological data, it's very high dimension though. So this particular data set here is, I think, around 3,000 by 3,000. I forget the exact dimensions. I think, so about 3,000 bone marrow gene cell counts by about 3,000 genes. So you have these really big data matrices that we want to understand. And there are many different ways of understanding a data set. One of them is to stare at it. But unfortunately, staring at this data set in this matrix form doesn't really give you really great insights. So, of course, the standard tool people use, and we've seen multiple talks talking about dimensionality reduction. So you might use something like PCA. And what PCA does is you have your big data cloud, and you do some rotation of your data to allow you to see the directions of greatest variance. So in this case here, you probably cannot see my mouse. Actually, no, you can't see my mouse. Great. So you might have this direction that has higher variance, and this direction would be PC2. So you all probably already know this. But how do you interpret these PCAs? Well, I mean, each of these new axes of your PCs is a linear combination of your original feature space. So yeah, you could go back and say, oh, well, if something's going along PC1, then these are the variables that are being hit. If you're going along PC2, these are the variables that are being hit, and so on. And there are other variants of PCA, sparse PCA in particular, that are easier to interpret because they only invoke a limited number of the original features. Okay, so since we have a linear rotation, we're able to, if your actual data is planar, you could rotate it and you'd see it all in your 2D PCs. So this would be PC1, this would be PC2. So it's a linear transformation. It works well at finding important lower dimensional planes that you might care about. But real data doesn't look like a uniform plane sitting in higher dimensional space. It looks a little bit more like this cartoon here. And so, but one of the things that we have found is that high-dimensional biological data often lives close to these low-dimensional manifolds. And so you can think of this a couple of different ways. One way, maybe in an evolutionary tree, if you're interested in phylogenetics, well, you have this natural one-dimensional tree-like structure that genomes are evolving along. And this tends to be true in a number of evolving processes. So we have this data that appears to live close to a low-dimensional manifold, and we'd like to understand what happens. So this here is a cartoon. But this here is not a cartoon. This is actual data from, let's see, this is a data set where you have 447 cells by data points by 25,000 or so features. And this is RNA-seq, so single-cell RNA-seq trying to reveal cell differentiation. And this is a prior study by Yang et al. In 2017 that showed that, well, if you do a PCA, you can see, okay, so these are different cell stages. These different hepatoblasts differentiate into hepatocytes going to the right in PC1. And if they start going upward in this direction, they differentiate into cholangiocytes. So you can see that this happens. And so you can visually, when you visualize this data, you're like, okay, great. There is some pattern of gene expression that causes differentiation. We already know this. And of course, you don't have to just use a PCA. There are other techniques you can use, these nonlinear dimensionality embeddings like t-SNE or UMAP, some of the popular ones, which allow you to really visualize how these points are situated in some kind of low-dimensional embedding. So this is the same data as before. It's a little bit harder to see since the dots are a little smaller, but you still have your class.
I screwed up when I did it. I'm sorry. 1 second.
Oh, yep. Well, technical difficulties. In any case, the basic idea here that we care about is whether or not is really trying to understand what it means to go along a trajectory in your embedded space. And that's what we're going to be dealing with in a, oh, wow.
I'm fixing it, don't worry.
Yeah, yeah.
Slow, and then slides.
Yep, great. Okay, moving on from there. So you can see that in this particular simple example, the t-SNE and the UMAP both sort of separate out the different classes. So these are each of the different stages of cell differentiation that we saw a moment ago in the PCA. In more complicated examples, you may not see the same behavior because PCA isn't powerful enough to capture nonlinear relationships typically. Okay, so that's all that. This brings us to trying to understand what's actually going on, though. So you could take everything from this first cell stage and say, okay, well, what's the difference between this cluster of cells and this cluster of cells here? versus this cluster of cells, and so on. And so you can do these sorts of pairwise comparisons. You just look at, oh, well, these are the particular genes that are differentially expressed between these two particular groups, and that's a very standard analysis people do. What we were interested in is trying to understand entire trajectories, being able to build a tool to quickly visualize the differences, the important variables, the important features along an entire trajectory, perhaps along some sort of thing like pseudotime here. So the basic idea is we have some high-dimensional data that's living up in some space up there, and we embed it down somehow. And you notice that this embedding actually isn't very good. So these green and yellow points here, they were all together here, but they've been split across. But no matter. So when you do a low-dimensional embedding, things don't always work out, but you do have some low-dimensional set thing that you can visualize. And hopefully this low-dimensional thing that you can visualize has interesting patterns that humans can see. That's why we put these pretty pictures in all of our papers, because we hope that those low-dimensional embeddings, t-SNEs, UMAPs, PCAs, those must evoke something. And so in this case, but what do they actually tell you? So you have a bunch of points and you're like, okay, well, they sort of cluster, and that's all well and good. And often these things are put up in papers or on presentations. You just say, oh yeah, the thing that we did, it sort of makes sense because our classes sort of roughly separate. Okay, so what we're going to do, though, is we're going to take this low-dimensional embedding. We're going to say, well, I, as a biologist, I care about this particular trajectory of cells. Maybe this labeling of pink and magenta cells has some importance to the study I'm doing. And I want to know what features are important. What genes are important along these particular data points as opposed to all the other ones. And so I'm going to zoom in. And whenever you do this, I'm going to only look at these cells. And our strategy is going to be, well, these cells, they correspond to something in that higher dimensional space. So you can back project them up to this higher dimensional space with all their features, all 25,000 features, or in this case, 3 features since we're visualizing in 3D, and just look at those cells there. So we subsection the original space that corresponds to the user-drawn manifold. So a user draws something, and it's like, then you're like, okay, well, what is important for that particular subset of points? Okay, well, since you've now projected back up into this higher dimensional space, you can look at the behavior of the original features along those cells. So you've now drawn a trajectory, and now you can say, okay, well, along that trajectory, well, how do all the other features vary? And so here I have 3 toy features. Maybe they go up and down in some particular pattern. And you can say, well, maybe a feature that varies in a particular way along your path is representative of that path. The sort of obvious case is, suppose you have some feature that just goes up as you go along your path, and you're like, oh, well, this feature, this gene, maybe the expression level goes up as the differentiation process continues, or goes down. But there are other patterns that might be important. Like if you go up and down, that might say something about the particular trajectory you're taking in your phase space. Okay, and so that brings us to SlowMoMan, which is the tool that we built. You can go to the GitHub link. You can play around with it right now. This is available. We'll see. I'll be demonstrating it in a moment as well. The basic idea is we allow users to draw these paths, and then we tell people what features are important. Now, that's a little bit complicated, of course, because how do you tell that a feature is important? And how do you do that quickly? Well, we need a way of ranking the features. There are lots of different ways of ranking features. This is something that's been done a lot. But what's a way that's quick that you can do in a browser in this sort of interactive manner? Initially, some people, and so whatever these importance metrics, we have several of them, you rank these features, and then that you can see what's important and what's not. Okay, we originally considered using variance of the sample because this is, of course, what PCA is sort of optimizing for. But this can't actually disambiguate between variables that are super noisy versus ones that are meaningful across a path. Because here, instead of just having a cloud of points, we have points that are along a linear path. And so we want to include that sort of linearity information, that sort of temporal information in our analysis of feature importance. Another thing that we could do is we could measure all the correlations. That measures the correlation of a variable with a time-delayed version of itself, which works a little bit better, but the problem is you need to guess the delay time, which depends on how long a path is, how long you draw the path, and like exactly the path you take. Instead, what we did is we realized that we will need something really fast, and we need something that captures different scales of interesting behavior along a path. And so we instead turned to the fast Fourier transform. So if you do a Fourier transform, what you get is you decompose your original signal into a sum of sinusoids. And then all we care about is that these slow motions, so the slow up and downs, like that there's some slow variance across the path that you care about, which is where we have the name SlowMoMan, so slow motions across manifolds. And the idea is we're going to emphasize the slowly varying features because those are most likely to be explanatory of whatever path it is you've chosen. So more precisely, we take a harmonic sum of the FFT amplitudes. Okay, and I think at this point, I am going to jump into live demo mode. So feel free to shout at me any questions if anything's confusing so far, but let's switch into this live demo. Okay. So I've already preloaded the data here. This data here is actually exactly the same as this data. So I've just preloaded the hepatoblast differentiation data into this. The colors don't exactly match, but that should be all right. I'm going to erase my line. And so, sorry, the interface is a little weird because the resolution on the screen isn't great. But the basic idea is you're able to load any 2D embedding. So you can load the PCA into this, you can load the UMAP into this or a TSNE, and then you can draw an arbitrary trajectory that you're interested in. So this one here. You compute all the variables by importance. Here we're using the FFT metric. And after this is done, you'll see that you have some variables that are going up as the along the trajectory. So this here, this path that we chose here, that path here is simply for differentiation of blast into hepatocytes. And some variables are going up and some variables are going down. So this is the most basic pattern that you might hope for. And you can see, oh, well, ApoH, which is, I am not much of a biologist, but this is apparently a apolipoprotein H. It binds to various kinds of negatively charged substances. This protein appears to be, this gene appears to be expressed primarily in hepatocytes, but not in hepatoblasts. And you can see this for, so this is a heat shock protein that apparently is highly expressed in the progenitor cell, but not as you do the differentiation happens. And so you can see that this path that I chose can be explained by a combination of these features. So these genes seem to be what is important for this particular path that we've chosen in the PCA. And what we're actually doing, if you zoom in a little bit, which I am here on the PowerPoint, you can see that we're just picking up points from the PCA, projecting them back up into this higher dimensional space, putting them along the line, and computing the fast Fourier transform of each of the features along that line. And so you may want to choose a slightly different path. So maybe instead of doing that, you want to do something a little bit wavier. Because you want to just pick up more points. That's all you're doing here, which is you're just picking points that you want to be representative. And if we compute that, sorry, I just clicked autocorrelation, so that is not going to work as well. We do have implemented autocorrelation and variance instead. So you'll see that you get many of the same gene expressions from doing this sort of wavier path, because of course everything is renormalized by the sort of fast Fourier transform that we're doing on the data. And so you still get many of these, although you do get slightly different things depending on the path you've chosen. Okay, so that's one thing you can do. So the other thing is you can take the other path. So everything we did a moment ago, you could just do because everything was along the first principal component, right? So it's like, okay, well, those are the variables that define the first principal component. That's not too interesting. But you can draw other paths, including this path where you're going along PC1 and then going up to PC2. So this is a little bit more difficult to interpret. You still could do it in the PCA, but it's a little bit harder because you're sort of turning. And if you do list now, let's erase the line. You can go up and compute what the important features are, what the important genes are for this particular path of the differentiation into cholangiocytes. And you can see some of the same things appear because, so like ApoH still appears, you still have there's some amount of overlap, but not complete overlap. So this is sort of following along cell differentiation pseudotime. But naturally, you don't have to go along this sort of positive direction of time because you can draw arbitrary trajectories. You can go instead from cholangiocytes all the way to hepatocytes and just be like, oh, well, what differentiates these two classes? And so instead of going backwards in time, you're just going sort of backwards and then forwards again along this trajectory in your PCA. And you can do the same kind of thing in the UMAP. So this here, I think, is the, yes, this is the UMAP of the same data set, and so you can do the same kind of thing and see which features show up as important in the UMAP or some other embedding. There are also really funny things you can do, because so like the sort of point of what we've been talking about is that finding important features along paths is more interesting sometimes than finding just clusters. And this goes back to the keynote earlier today where they were talking about, well, whether or not there are specific clustered states or whether there's this sort of continuous range of differences between states. And so if you really do believe that there is a clustered state, so you could just draw a squiggle here and a squiggle there, and all it's going to tell you is the difference between that particular group of points and this particular group. Because, of course, when you compute the FFT of this, what's going to hopefully end up happening is, well, some things are only in one cluster and some things are in the other. This is not the way I'd recommend doing this, but you can sort of see that even if you draw these sort of arbitrary non-continuous paths, you can still see what the difference is between the two different sides, because the FFT is capturing that. Since when you're taking the harmonic sum of the FFT, these motions that are down for half and up for half, those tend to score very highly. Yeah, and you can also, it's not just the differentiation of things. You can also see things that, well, if you drew something that went down and then went back, you might end up seeing some variables that go down and up. Yeah, so you see some variables here go down and then up, which might be important if you have things that are regulated, well, downward and then upward in a particular trajectory like you've chosen. Okay, yeah, so that's basically it. Like, that's the idea behind what we're trying to do, is we're just trying to build a tool that so we built this originally because I wanted to be able to see some differences along trajectories, and I didn't just want to do a cluster by cluster analysis. But of course, you can recapitulate trajectories from other analyses. So in this particular case, Paul et al. Used something called PAGA to figure out important genes along a particular UMAP embedding. And we're able to recapitulate some of those by drawing a path that sort of follows along the paths that they found in terms of these cells. That's more details about that in the paper. Yeah, so the general use case is finding important features in embeddings. The other sort of fun thing you can do is, this doesn't just apply to low-dimensional embeddings like PCA or UMAP. This applies to any 2D map you can impose on your data. And sometimes it turns out that we have another natural 2D map of our data, especially with spatial transcriptomics. So this is some data from 10x. So you've probably seen this. This is some of the sort of base public data that a lot of people have been playing around with on lymph node tissue samples. Well, you can do a UMAP clustering, and you can do this sort of analysis on the UMAP. Or you could do this sort of analysis drawn on the original 2D location. So if you have some meaningful 2D coordinate system for your data, you can still figure out along paths in that 2D coordinate system what features are important. Yeah, so if there are other natural embeddings, such as in spatial transcriptomics, you just upload those instead. Let's see. Oh, yeah, the last thing is, let me switch over. We have some toy data sets that are already loaded. Here is a fashion MNIST data set. So this is not biological at all, but this is, you can see these are different images. You can sort of see up here of T-shirts versus blouses versus dresses, etc. That we've trained a neural network for, and we're trying to explain what which things in the neural network matter. And so to do that, what we're going to do is we're going to draw a path through some of these things, and then, oh, I drew a bad path there. Let's erase that. And then what you can do is you can see the important neuron activations to explain different things along that path. So these labels aren't that meaningful because these are just variable names, but you can see that you can explain different types of feature sets. So these are the last layers of activations in the convolutional neural net. And I think lastly, I'm going to throw in a human microbiome data set that I think is quite fun. So this is a T-stochastic neighbor embedding of microbiome samples taken from different parts of the body. And this is just all the features here, about 24,000 different OTUs. So the different bacterial species that are present. And you can follow along a path and see going from one part of the body to another and seeing how which bacteria change. So over here, I think I was starting, sorry, let's click that button. Ah, there. So can I zoom out? Okay, yeah, so here you can see that along this particular path that I've chosen that ended up here somewhere around the hard palate and the mouth. Well, over there, you see lots of streptococcus as part of the bacteria that's present in that sort of sample. And you can sort of see the differences and along these paths that you've chosen in your t-SNE, which different bacteria seem to be explanatory of where you are. So everything that I've just been doing has been running on this machine here. So this is a live demo. It's running in pure client-side JavaScript on whatever machine you're using. It doesn't have to do any server-side computations. The reason we were able to do this is because, of course, the FFT is super fast. We have two different versions of the software. We have one version that has nice features like being able to change the sizes of the points, etc., and select things. And we also have another older bare bones version that is slightly less featureful, but is slightly faster as well for doing this. But yeah, as you can see on these data sets, like the amount of time it takes tends to be around a minute at most to load the data, and actually exploring the different trajectories you may care about, that tends to be a few seconds at most. Yeah, I think I will conclude. So nonlinear low-dimensional embeddings are powerful, but they're hard to interpret. We're trying to help you interpret these sorts of data. So we want you to be able to, if you notice something cool in the low-dimensional embedding, I want you to be able to just pick out that embedding, pick out that particular trajectory and say, oh, well, these are the features that we care about along that embedding. We're able to do this fairly quickly because we're using this harmonic sum FFT score, which I don't think I've seen elsewhere. Like, it seems like a very natural thing, so I'm not sure if anyone else has used this for trajectory feature importance. There are other types of feature importances that you may care about, some sort of regression analysis and that kind of stuff. We found that was too heavy to put into JavaScript on your web browser. But those are certainly things that, if the sort of trajectory analysis becomes more important, one might want to look into. Yeah, I think that's about it. The code is available there on the website. You can also go to the QR code. And I'd like to acknowledge especially Karen Dio, who made everything look much, much prettier. So I wrote a really ugly version 1 of some of this code like several years ago, and Karen made it actually look nice and usable. Griffin Weber, one of my collaborators, and my lab at Carnegie Mellon, where I am now, as well as a lot of my lab members back at University of Toronto, including, wait, did I not put Jim on here? I think I originally had a picture of Jim. Okay, anyway, I forgot Jim, who is in the audience, but he gave a talk earlier today on SILF. So thank you all so much. If you have any questions, please ask.
So I have a question, maybe two.
Yeah.
So one, I see that there's line width. Does that just change which cells, like, around, or which pixels?
No, this is purely visual. So basically, right now our proxy is at every point along the line. We just choose the nearest real point.
Okay.
Yeah.
So then that leads it to sort of a secondary question. Can you add, like, would it be possible, or like, is it useful to add sort of like blobbiness to it? Like, I want to select around here, and then, you know, whatever the trajectory, and then all of these guys here.
Yeah, I think it would be just because like you may see that trajectory that you might not capture it exactly by drawing over it. Right now, the thing we've been doing when we've been playing with it is drawing these sorts of wavy lines, but that is a bad way of doing what you're suggesting. So yes, that is absolutely something that we could incorporate in there. There's a little bit of complication when you have really, really busy t-SNEs, because like sometimes that sort of blobbiness captures a different like branch. Which is a problem when you have a lot of data points. But for these smaller number of data points, absolutely.
Yeah, no, I don't know if that's the implementation, but it might be useful.
Yeah.
Two questions, I think, or maybe one of them, why is that a mathematical form of the other? I'm really excited about the idea of the tools that would help me understand my data. And on the other hand, I have no idea how I would choose which trajectory to draw. And I mean, it seems like this is sort of saying I can arbitrarily choose a set of things, which I can't really justify. And then I'm going to give you both questions once. My wise-ass question is, does it recalculate the false discovery rate for everything you draw on the same data set? Because you're essentially doing more tests.
Yes. So that is one of the reasons why we don't actually have a false discovery rate thing on here. We think of this mostly as exploratory. So like if you're interested in just seeing what variables seem like they're important, but we very specifically don't try to give, we just give a relative ranking and not an actual, like, this variable is explanatory, because I don't think we can with like the sort of arbitrariness of the paths that people draw. In terms of what paths you would want to draw, that depends on your data set. And sometimes people will have the sort of, in this particular case where your data is already labeled, you may want to draw a path because you might want to see, oh, well, if you go from saliva to tongue, like, or I mean, that's going to be pretty similar, but all the way to, let's say, the retroauricular crease, like, what are the differences as you go along that path? And yeah.
Why wouldn't you just say choose the things with these labels and tell me what the,
Because the idea is you might think that so this goes back to the question that was raised in the keynote. In certain cases, like, you may think that your data is actually categorical, but are the microbiome samples behind your ear as you go down into your mouth? Is that a like step function, or is that a continuous change? And if it's a continuous change, then doing a trajectory is going to give you something a little bit more fine grained than just doing a 2-cluster analysis. If on the
we're banking on the idea that new map or something like that
is capturing something real. Yeah.
turns a categorical variable into, because when I sampled it, I said it was categorical.
Yes. So you have a categorical label for your info. Yes.
I have the location.
Yes. So
And I hope that the trajectory also has location.
yes, you may. Yeah. Yeah. And so, like I said, it really depends on like, should you ever trust a UMAP? Well, I mean, that's a broader philosophical question. Yeah. 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 And so, like, what I guess, like, what this is doing is this is allowing you to better interrogate like what the UMAP is actually showing you. And so. Yeah.
Was there another question here?
Yeah, I was purely out of curiosity, like, how many features is too many features?
So right now, so this version of the software is a little bit less efficient than the one without the fancy features. For the other version, like on my laptop, which is actually less performant than this one, you can probably get up to around 30 to 40,000 features and 100,000 data points. So on a bigger laptop, you could do more than that, but certainly like that takes around on the order of like 30 seconds to load and like 10 or so seconds to compute. It scales linearly with the number of features. So
if we apply it to Kush's data, will we find out what the difference is between what his model has discovered and what was in the model that explicitly looked at dielectric function? So
I don't know. I think you'd have to do yeah. If you send me the properly formatted CSVs, I could throw them in, I guess.